The ecological significance of mint plants aligns closely with our research on the impacts of resource availability and climate change on plant health and ecosystem dynamics. While mint plants, like other members of the Lamiaceae family, support pollinators and enhance biodiversity, they too are affected by changing environmental conditions. Our research explores the relationship between research limitations, guided by the law of the minimum and law of the maximum, and plant health, specifically focusing on how variations in soil quality affect growth and resilience. By studying the response of mint plants to manipulated soil conditions, we can better understand how critical nutrients influence their ability to thrive and support pollinators in a changing climate. Linking our findings to the broader role of the mint family in ecosystems provide a practical application for the research. Determining where mint plants thrive better will help us understand their ecological values, which means the support they provide for smaller organisms that influences the bigger picture. This knowledge could inform strategies for creating pollinator-friendly habitats, helping to mitigate the negative effects of climate change.
The groups consisted of control, extreme, and optimal. The control group only received the topsoil for all 14 containers, while the extreme group had an increment of 1/4 of teaspoon up to 14 that corresponded to the 14 containers, and the optimal group had similar but an increment of 1 pellet of nitrogen. To test our hypothesis, we conducted a control experiment with 11 brown plants. Over the 5-week period, we manipulated nitrogen level between the groups and monitored growth while maintaining similar precipitation and UV light by periodically giving a similar amount of water and swapping the location of trays to account for natural UV radiation cycles. We collected our data on plant germination, stem height, and numbers of leaves every 2 to 3 days to see the growth over time, and directly inputted these data into Excel for better efficiency. Although we weren't able to be consistent with the days of collecting data, our data still provides the growth over time, allowing us to observe the differences as time goes by. Throughout. Meanwhile, for measuring germinations and leaf, these were simple because it was only a matter of counting. Surprisingly, there was a mutant plant with 3 sets of leaf instead of the normal amount of 2 that was doing as well as others. By the end of this experiment, we observed differences in average mint biomass between the control and optimal groups. But since our standard deviation was so close to each other, it suggests other factors may have influenced our result.
However, for the average number of leaves in mint between the two groups, our data indeed supports the optimal group to be more favorable, since resources required for leaves for production are costly, which allow more photosynthetic ability to create more energy that can be used for further growth, meaning the sugar created by plants can be consumed by animals for their growth.
As for the growth of length and germination over time, it seems to be same among these groups, which can be explained by the similar growing factors that were present in the greenhouse.
Although, this result suggests more time is needed for better conclusiveness and experimental design. What we know from this experiment is that the law of maximum for sure exists, and don't let marketing tell you otherwise. That is only use what's needed.